poster. So Michelle is our partner at Charles River, and this IFAX works on single cell necrosis and other findings in the liver. So yes, please tell me how we did it.
Okay, so this is our first object-based detection classifier. Previously we've done a lot of CNN-based work, but we wanted to go into the world of single-cell events. So we had a look at necrotic and apoptotic cells within the liver, as well as the mitotic cells as well. So we had a data set of about 1,200 examples of both cell types, and then across 26 studies and a number of different sites and scanner types. And then what we did was we looked at two different classifiers. So one with just the two classes of mitotic cells and necrotic apoptotic cells, and then one with this additional other class, which was created to filter out artifacts and other different types of cells like Kupffer cells that we were having a look at when we were training this and kind of evaluating the classifier. So we did a number of different evaluation processes. We had a look and evaluated based on unseen tiles, then we had a look at unseen slides, and then unseen studies, and then eventually we would kind of have a look at the intended purpose of the classifier and make sure that it's kind of aligned with the metadata that we were seeing from the study output.
So question, why did we do 2 classes and 3 classes classifiers?
Yeah, so the initial, results were showing that we were getting a lot of false positives on the likes of artifacts and other cell types. So that was why we tried to add this additional class in. But ultimately we found in the results that it performed better overall when we didn't include this class. Mm-hmm. Yeah, so we were initially going to not show the other class and just have the 2 classes, but ultimately it worked out better just to have the 2 classes that we were trying to identify in there.
So what's the plan for the future for this particular classifier?
Yeah, so it's still in iterative improvements, and the very good AI research team at the IFAX and Charles River are still doing a lot of work on this classifier. So at the moment we're still just trying to improve the data set that we're using and make sure that we have a large, diversity and variety within that data set. We can never have enough data on our side, but
you can never have enough images to train on, right?
Yeah, so then what some of the things that we're having a look at is kind of isolating out the hepatocytes from the sinusoidal regions as well. Yeah, and then as well then we're having a bit of a look at the UI as well. So we have this density-based view that we can show within the study browser on Pathologics, which means that you can see the hotspots where the highlights is of kind of any single cell events. Working on the UI alongside the classifier as well is what we're trying to do there to make sure that it's an improved workflow for the toxicology pathologist.
I am looking forward to this one because we also have another poster here from the IFAX, and this is something that many pathologists at Charles River were working on. And this is development of different classifiers. So those single cell findings, this is just one, right? And it had to be done in a different way. That's why we have a separate poster on that. Whereas all these other classifiers that we just recently tested were done just with classical supervised deep learning. And let me show you which organs they were designed for. Here we have for heart, lung, kidney, thymus, adrenals, liver, thyroid, and this is gonna be our next iteration for the other organs. And we just finished evaluating this, several Charles River pathologists, and I started the evaluation with very critical mind, and I was positively surprised. I was faster and I was more consistent. So yeah, it was cool because it was like several years of work. How many? 3 at least?
Exactly. I think we probably, it was 3 years of work overall. I think it was everyone's probably first toe in the ocean in terms of this. So we learned a lot along the way, and now we're kind of it's kind of that the cogs are in motion so that we could produce any classifier for any organ now, for any species, because we have this setup available to us.
Yes, I liked it a lot.